for Paulucci Award. He had like 500 some publication, 80 students, and now he's almost maybe already exceed 700 publications and 100 students. So just put into perspective what that means. At Yale, you can get tenure with 100 perhaps publications. So he can get tenure for 7 times at Yale. Okay. So I guess she's not ready yet. So he was, again, my junior colleagues. And I guess it's also good concrete examples to, sorry, put you in that spot again. So someone who worked on important problems, and as Qi-Hung corrected me, try to change the world and make it better world. And consequently, also were rewarded for many awards and recognitions. And we are proud of Hongyu for his work and the leadership at Yale. Are you ready? Okay.
All right. Thank you, Heping, for a very kind introduction, and also thank Tian Hong to bring me here. We really have a great time, Abu Dhabi. I think Qi-Hung already gave a very good introduction about the topic that I'm going to talk about, and also allude to a little bit, but in Hong Zhao's talk about prediction. So this is not too long or after people start getting some ideas of genetic contributions to complex traits. People start asking questions about the disease risk for different kinds of phenotypes that we are interested in, like income, education, who are going to marry, how soon or how late you're going to die, etc. A key question in this genetic risk prediction is purely based on genetics. Like we have, Qi-Hung mentioned, we have billions or variants. In terms of common variants, we have millions of variants. How to score them, how to combine the genotype from million sites to really come out with a good model. The question is what markers to choose and how to score them. It's very simple. The other thing is, I also mentioned earlier, is a lot of GWAS, genome-wide association studies, only provide summary stat. So instead of having individual level like training data, like we need to handle the case where we only know the marginal significance levels. And because the question is very, very intriguing, right? Everyone wants to know the predictions, right? Want to know how the health is, etc. So there's a lot of interest activities in this space. So this is a review paper done a few years ago by Zhou Xiang at Michigan and his students. As you can see, going from left to right, there's more and more activities, and also the color changes from blue to yellow because the later one, the yellow ones represent these methods only based on summary stat because there's the availability of the data are there in this space, and including some of our methods here, EBPRS, and like the SDPR, almost toward the end that we have some methods. And you look at the different methods, and they are all pretty much based on these very simple linear models. Like, it's not as simple as what Qi-Hung talked about because there may be a million predictors here instead of just like dozens or hundreds of thousands. And so all these may be very, very simple model. And but the key differences among different methods essentially lies in how to really specify the prior distribution for the effect size. And the next slide shows the different varieties. But the question is which one really represents biological truth? Nobody knows. And people try different methods to see which ones that really can give you good prediction result that can give you something about which you feel more comfortable with. So these are the earlier ones that simple like point mixture model, point normal mixture models here. We can keep adding the normal components, and so there's one method by us which has put the extreme, to include like 0.0 plus many like normal mixtures, and it seems to work pretty well. Computationally, it's also feasible, and also this is more based on the full Bayesian, also empirical Bayes. So just said, the points here is all based on like a lot of different ways to specify priors and different ways to estimate. Whenever for most time I give this talk, people ask me like, look, all these methods are great, but all is based on very simple methods, right? And we have this great method called deep learning. Can you do better than these simple methods? And the answer in this context, we tried, and several students persuade me to try. I said, great, and none of them could come up with a better result than these simple linear additive models. that's a very simple way to think about the problem, like the linear models, but also there's a biological context here. As Xihong mentioned in her talk, there's a lot of different ways to annotate the human genome. And this annotation, this functional information layer actually can be quite helpful not only in identifying variants associated with the traits, but also in predictions. And also there's a lot of shared genetic effects across populations, also across traits. And both kinds of information have been utilized to improve prediction results. So this is a figure just to show that comparison. That this is a paper under revision by us. Look at the first, the first one here is GWAP anno. Anno means GWAP is a method. Anno means we use annotation information layer. Just look at this one with the reference. There's a very common used called PRSCS from Boston. This is the reference one. The number positive means doing better job. But pretty much by using annotation information, you can actually improve for different traits, like this asthma, breast cancer, IBD that Hongyu Zhao mentioned in his talk, coronary artery disease, type 2 diabetes, schizophrenia, etc. That's for quantity for binary traits, but also we can do better for quantitative traits like body mass index, etc. Right? So using annotation information layer helps. The other thing that's also using shared genetics across populations, across traits. Also, Hongyu Zhao mentioned in his talk about transfer learning. This is very different kind. I mean, this whole thing about transfer learning, transferring what you know from European samples to other non-European samples, because for most genome-wide association studies, they have been done in European samples. Therefore, there's more information to estimate effect size for different variants. As you move away from Europeans, especially in Africans, you see the performance actually deteriorates pretty rapidly. So in this case, you really the bottleneck for African population is a limited sample size. Like for in Hongyu Zhao's case, a limited pen match sample size. So it's a lot of you can really borrow information from these larger European samples to improve the estimation effect size for the non-Europeans and these samples. And we have a paper that's under revision here, under review right now called the joint PRS. Actually trying to borrow information to the question of how you specify the share effects. This is obvious. There's a little technical details, but also we can want to make a computationally feasible. And we can see that for like different populations, East Asians, Africans, and these are South Asians, and our methods are actually, the positive number means that our method, joint PRS, is doing better job than the other methods. And right now people a lot of time use something called PRSCS, X, cross-population, we are doing better job. There's other methods. Some include method developed by us, but in general, joint PRS seem to have a better way of characterizing, characterizing share effects, also computationally to do a better job. Now come to the disease risk prediction. So far, I have talked about it's very simple in a way that we only use genetic information there, but we know a lot of other information, like for example, the age, the other demographical variables and clinical variables are also important. And people have paid relatively less information there, less attention to how to combine genetic, clinical, and demographic information there. In this space, I cannot really, cannot say confident that the AI cannot do a better job. Actually, it may do a better job if we can really get some way to summarize the genetic contribution there. That may lead us to better prediction. And another thing that people haven't quite utilized, in Hongyu Zhao's talk, you can see there's prediction over time, but actually a lot of phenotypes like UK Biobank, the EHR, you had observations over time, but that information has not been quite utilized well in the current prediction models. And the other thing also mentioned by Xihong and also Hongyu Zhao is there's a lot of rare variants in terms of whole exome sequencing or whole genome sequencing. How to really capitalize on this information layer, there's very, very few methods utilize that information layer. Multi-omics, mentioned multiple times. Imaging, the first session, exposure, like the exposure, the temperature, etc. That information hasn't been utilized. If you have asthma, some basic problem, if you have really poor like air quality, it's problematic, and other data sets. And as already mentioned, the deep learning methods actually failed to compete with very simple like additive models for genetic risk prediction, but that may change when we consider as the clinical information, other information there. Actually, this is a very large open space. in line with the previous lightning talks, also with Martin, I try to answer the questions given to us, some by Tian and Hongyu. So what would be the next stage for research in this field? Again, we want to really get the more accurate disease risk prediction models, not only using genetic, but many other types of information there that we have access to, some with complete information, but some with incomplete, like Annie talked about. Like, there's a lot of data, we may not have complete information there, right? And also, we really want to convince people, people still are questionable about this, how useful the PRS, these things, but successful clinical trials, there's several ongoing right now. I think she was involved in one in the VA, to really demonstrate the usefulness of PRS, but more generally, the risk prediction can really help to stratify people for early cancer, like for detection as well as the cancer monitoring. And also, what one will go into is really to see these things get adopted in the different healthcare systems, right? And to stratify people into different risk groups and to improve disease prevention and treatment. And also what would be even better, we can have something like Apple Watch, right? You can get into the thing, you can have monitor there to give some people warning, like today it's really warm, probably just stay inside, etc. Yeah, you can see the irregular heartbeat, etc. There's all this information there that can set up a warning. This could be really useful. What will be the most exciting pivotal breakthrough that AI and stat could lead to that will change the practice? I think this has to do with a lot of the sharing of bank level data, because so far all these methods have been based on summary stat. There's enough data to show that if you really have individual level data, you actually can get much more improved prediction accuracy, as you can imagine. There's two things that have been the bottleneck and the roadblock. One is the privacy issue, the other one is the computational issue. I think both can be addressed, hopefully. And also, we really want to look at dynamic models. My risk for disease may be different for today from tomorrow, from a month from now. Maybe it's different in the summer or winter, and that thing hasn't been really considered very seriously. Yeah, and the other thing has been mentioned multiple times, how to quantify uncertainty in risk prediction. The most important open question that the research community is grappling with, I think right now there's a lot of interest looking at the cross-population because the health disparity issue there. But I think this will probably be done, I hope, within the next couple of years, and people can really work on the more important thing is how to get these more comprehensive models that incorporate different risk factors and complex interactions. This is the place where probably the AI can play a major role instead of just a simple linear models here. How could individual statistician get involved? I think this is the same answer. I mean, we are talking about the same, so I just saved everyone's time. We're just getting involved in interesting problems, be part of the team, and to really contribute to science instead of really thinking about just getting published in these JASA other journals. All right, thank you.
So I also want to thank Tian to give us 15 more minutes so that we can discuss and ask questions for any of the speakers we have so far.